but I have a question for you. In your experience, have you found short shelf life of viral vectors to be an issue? Yes or no? And if so, what advice do you have for our audience members today who might be struggling with the same issue?
I'm sorry,
Hi,
was that
Curran.
for me?
Oh,
I had a
sorry.
lag,
Yes, yes.
I had a lag in the screen. So was the
Not
question
a problem at
around
all.
shelf
So short
stability?
shelf life of viral vectors. Yes. Yeah. Um,
Yeah,
have you found it to be an issue? And if so, just some advice for our audience members who might be struggling with the same thing.
yeah, we don't see too many problems. Of course, the product is stored at below -60, and so not a lot goes on there. The timing for we don't have stability trends or issues that we see with AAV8 or 9 with good formulations around them. So we project for bulk drug easily a 5-year shelf life. We are looking at for indications where storage at that temperature is highly undesirable, 2 to 3 weeks at a higher temperature, maybe 2 to 8 even, at the clinical site so that they don't have to have a -80 freezer on site. And at those temperatures, we do see a more narrow stability for the product, but that's managed through the supply chain pretty easily. So in terms of long-term stability, either of drug substance or drug product, this is not something we are too worried about, even though we have 3 different formulations we use for 3 different indication areas.
Yeah, sure. That makes a lot of sense. Dave, I want to make sure I ask the same question of you, especially of, all the other vectors. How have you found short shelf life to be an issue? Have you? And if so, what could you share with our audience on that?
Yeah, well, I mean, I agree that shelf life is generally measured in at least from an industry practices standpoint, long-term frozen stability, maybe some amount of 2 to 8 or what I call refrigerated stability and room temperature stability. As you might imagine, most vectors tend to have a decreasing correlation as you go from frozen to room temperature in terms of the length of stability they have. And within the vectors that I've had experience with, AAV tends to be quite stable for a number of years, as do several others, adeno as well. Whereas things like lenti and maybe the enveloped viruses, if you will, tend to be a little less stable. I think they have more complex surface chemistry and biochemistry that can render them a little less stable. There is there's an entire field of formulation development with a subfield, I should say, within our industry that focuses on coming up with the best recipes, if you will, for both formulation buffers as well as any other excipients and whatnot that can be added. And it's worth the spend to do that, to put the effort into coming up with the best possible formulation that you can, because once it's out of your hands at the healthcare providers and whatnot, there's never any guarantee or telling exactly how they're treating your product and whatnot. And if it's sitting around on a bench or something for longer than it should, a really strong formulation can, help bail you out of a couple of circumstances that you otherwise may not even be aware of, as long as you're not losing the stability. So it's an important aspect for sure.
Yeah, sure. As you can see, the results of our most recent poll question, one of the, I think, tied for first, the greatest challenge in viral vector development is, of course, scalability. And so I want to talk a little bit about how the challenges associated with single-use technologies as businesses scale their viral vector manufacturing. So when it comes to single-use technologies, talk us through a little bit about what we can be doing a little bit better. Curran, can we go to you for that one?
Sure. Well, the number one issue over the last 3 years was getting bags. So I think we're a little bit past that now, but you can see for single use for the upstream process that the supply chain really wasn't as robust as we wanted it to be through the COVID period. So there was like I said, a 2 to 3 year period where we were wondering if we could even run. And I did hear people saying, gee, the old stainless steel tanks never had those issues. So I think that's a challenge that the suppliers need to now look back at that and say, how do we prevent that if we have another pandemic or other type of issue, so that the supply of single-use bags is really something we can all count on and know, especially when you're starting to do conformance lots and commercial lots. You don't have the flexibility to change equipment anymore. That's really going to be a key risk that we evaluate as part of a commercialization process. But having said all of that, single-use technology, as I mentioned earlier, is allowing companies to start up manufacturing in-house or at a CMO within a couple of years of building the facility. And time is the most precious thing that we have to work with, that anytime we can pull those kind of timelines in and establish capacity and establish the lack of having to do hundreds of steam in place and clean in place cycles that were really wasteful and expensive in the old days. It's just been a really great development that's kind of fully materialized now. We are worried a little bit about all of the consolidation that's going on from those that supply equipment. And I'm talking about large scale GMP equipment that, how is this all going to resolve over time, given that we've now interfaced this equipment with all of our software in the plant, and we've interfaced it with remote workers who are watching batch processes. If some of that equipment gets obsoleted because of a merger of multiple companies, then that's something we'd have to redo, and we're just concerned about some of the M&A that goes on the vendor side.
Yeah, sure. Thank you. Dave, I want to be able to hear from you too. When it comes to single-use technologies, are you feeling that businesses can scale their viral vector manufacturing? I'm sure you concur, of course, with a lot of what Curran has said, but I want to give you the opportunity to respond to.
Yeah, appreciate that. There's no doubt. I don't think anyone would necessarily disagree that if you were to ask, poll 10 people and ask them why go with single-use technology, invariably you'll get a fairly common response around elimination of things like cleaning, validation, and all of that, CapEx infrastructure, if you will. It doesn't come without challenges. There's no doubt about that. Single-use technology is obviously a very large term. For years there have been situations where bags were, supply chain notwithstanding, and that has been a challenge for sure, as Curran pointed out. But additionally, even, when there aren't supply chain challenges, there have been some product quality challenges over the years with respect to some of that technology. If someone has worked with single-use technology for, over a year, they've probably experienced at least one critical bag leak or one failed tube weld or something like that, put the batch in peril, so to speak. Those are realities. They can also be, that sort of technology can also be super finicky and complex to use. Anyone that's ever used a 2,000 liter single-use bioreactor knows that bag doesn't just install itself. Those are complex pieces of equipment for sure. Again, a lot of upside. But I do think that, that said, I don't want to just paint a sort of bleak picture. There have been tremendous improvements, I think, in the technologies. And I have to applaud the supplier companies, the MilliporeSigmas, the Pauls, the Sartoriuses of the world that have done a tremendous amount of work and really spent the capital internally to next gen almost everything that's out there and being used. I mean, a few years ago, it was very, very difficult to get a batch end to end without some kind of catastrophic circumstance. But nowadays, I do think that that has been dramatically improved.
For sure. We'll add Thermo Fisher Scientific to that list too.